Good afternoon, everyone. So we keep to time. I'm going to start the session on time. Thank you all for coming to this afternoon session. We've got some interesting talks around seed enablement and technology, but also some genetics, which is from my PhD background, so I'm happy to see some genetic work along with seed work this afternoon. And first up we have Simon from the ARC Mine Site Restoration Centre. Thank you, Simon.
Time here. All right, thank you, Alison. So yes, I'm at the Centre for Mine Site Restoration, based in Perth. But on a previous life, I used to work in Europe. I'm originally from Italy, and along with a colleague, we actually started the first native seed company in Italy, and then we started collaborating with other companies across the continent on an European project. So that's actually what I'm going to talk about. It's about my European experience. Why is that relevant to Australia? Well, because we've been in Europe through some step of the process of developing a seed market, and maybe you in Australia can learn from some of our lessons and our mistakes. And as Paul Gibson-Roy is going to talk tomorrow about the situation in America, I hope that could be interesting for you to have a snapshot of what's going on in Europe. So as a general background, Europe is smaller than Australia, yet we have 20 times your population. And that density, along with hundreds, if not millennia, of disturbance, has resulted in almost no natural ecosystem left in the continent. What we call natural is actually semi-natural or cultural ecosystem, like grasslands or managed forest. It's a post-glacial system, so different to what you've seen before in the Pilbara. Natural regeneration occurs, but when it occurs, it tends to be taken over by invasive species, like it does here sometimes. And according to a recent assessment on biodiversity in Europe, it turns out that 65 of the important habitats are actually under unfavorable conservation status. And basic ecosystem services are deteriorating across the continent. And that's the need for ecological restoration. This restoration can happen within this legal framework. This is the called Habitat Directive, is the most important legislation in Europe for conservation. And alongside the so-called Bird Directive, that surprisingly was developed for protection of birds, has prescribed something like 27,000 protected sites within Europe for a total of 18% of the continent covered and protected within this legislation. According to the EU Biodiversity Strategy, Europe has committed to restore 15% of its degraded habitat within 2020. And to do so, well, there's a huge need for native seed. But what we are at native seeds, when we consider it from an academic or conservation point of view, we're very advanced. Like in 2000 was opened the Millennium Seed Bank, which probably most of you are aware of. And from that initiative then started the ENSCONET project, which is the project that set the standard in Europe for seed collection, creation, conservation protocols, and created this network of seed bank across the continent. And recently, this project called NASTEC, that ended last year, was actually trying to start engaging with the private tech sector and native seed companies to get more of a connection between academia and the industry. So we can see through time that we moved from the 2000 from a more restoration and conservation focus to a wider collaboration with the industry for production, broad-scale production of seeds. But what is this industry? Well, it started, probably the first place where it started was the UK around the 60s and 70s, where they are the first companies that start producing seeds of native species of local origin. Something has developed also in Switzerland and Austria, mostly for the conservation and restoration of grassland. But the most important case, the one I'm going to focus the most today, is Germany. It's the German example. In Germany, they started around the 80s. The first companies started around that time. And initially, they were very small productions, but then increased over time, both the production scale and the number of companies through the country. In the meanwhile, local government, even at national level, they start incentivizing and recommending the use of native seeds of local origin for revegetation, conservation, green infrastructure. For example, the one in the corner is the famous Allianz Arena in Munich, which is the stadium for Bayern Munich. And all the seeds outside it, all the fields around it, are all made with native seeds from a local company. Oh yeah, and this market, up to a few years ago, was actually unregulated. So they were selling the seeds, but there was no legislation in place to regulate or to guarantee the quality of seeds. Kind of, because there was a legislation that was developed in the 60s in the European Union, when it was still the European Economic Community, that was aimed to homogenize the different national legislation of seed qualities. And this specific directive is aimed at the fodder seed species. This legislation was aimed mostly to protect the consumers, the customers, the people that are going to buy the seeds, and guarantee a certain level of quality through labeling. And what this directive mandate for 24 species and 4 genera of grasses of legumes used for fodder is that every species, every variety that you sell needs to be registered in this special register and need to be homogeneous and stable. That means genetically very narrow, and also need to require minimum standards for germination and purity. This European legislation was then taken by the German government and embedded into their legislation called the Seed Traffic Act, or SATG. And it's very important because this is going to be part of the story later on. So we present the situation here. You see on one side you've got this growing market of native seed producer and lots of requests, lots of business starting here and there. And on the other side you've got the traditional plant variety breeders and the plant variety producers. They see a part of the market taken away from this side. So they decided to issue an injunction to stop the sale of seed of wild origin. In the case, they claim that these seeds of wild origin are not recognized and approved under the Seeds Act, and therefore they should not be traded because the native seed producers are trying to take advantage of this situation and are violating the norm. On the other end, the native seed producers replied that this kind of material, native seeds, should not be considered under this legislation because seeds for fodder species are meant to feed cows. Seeds for native species technically not meant to feed cows. We're meant to improve the biodiversity conservation status, therefore they need the genetic diversity. And the court ruling was that yes, those seeds are not admissible underneath this legislation. However, they recognize that this kind of material, the native seed materials, is not meant for agriculture but for conservation. Therefore, they decided not to issue the injunction, which was huge because if back then in 2003, this court would have ruled otherwise. Right now in Germany it would be illegal to sell native seeds, and creating the precedent that would make it legal all across Europe to sell and produce native seeds. So luckily they've decided this way, and the German native seed producers reacted kind of strongly to this because they were put in danger of their own existence, so they need to get organized and to need to prove seriously what actually native seed means, because at the time was still a bit up in the air. So to protect themselves from further attacks, they've decided to develop this seed zoning system with the help of lots of universities. This system is still up to debate, but today is a very good and practical example of a seed zoning system. And they've also organized in different associations the group's native seed producer. This one, BWW, is the main one. They also produce this seal that, for the quality and the origin of the seeds. And if you want to know more about the German Seed Association, I recommend you visit the INSA website, visit our webinars. At the very bottom of it, there's actually a 20-minute webinar by Anne-Karen Mainz, which is a representative of the VWW, that explains in detail how this system came to be and how it works. On the other end, we've got the native seed producer getting organized, but also the plant varieties were trying to fight back this new problem, and they do so by lobbying Brussels, enforcing this new directive, which provides certain derogation to the old variety directive. It could actually allow for the commercialization of native seeds, therefore making them somehow legal, but it puts stringent limitations on the labeling and quality specification that sometimes, or actually more often than not, are not applicable to the small batches of native seeds, and doesn't provide any specification about the origin and the provenance of the seed. So how this directive is written, I can actually collect my seeds in Italy, grow them in New Zealand, bring them back to Italy, and they'll be fine. But most important of all, the main limitation is actually about the quantities. They set this limit of 5% of the market that could be sold for native seeds. It means that out of 1,000 tons produced of fodder seeds, just 5,000 could be native seeds, which is not a problem in most countries, But it will be a problem very soon in Germany, because Germany has already got a big market, they've got more than 200 producers, and because of this legislation, or the Federal Nature Conservation Act, by 2020, in all natural areas and surroundings, it's mandatory to use native seeds of local origin. This means that the production is expected to grow 10 times and will definitely breach the 5% for the quota. And that's Germany case alone. If you look what's happening in the EU, we've got lots of different problems in different countries. In the UK, which is debatable if it's EU or not, but for now it's still there, they have a different problem. So somehow the customers don't expect the quality in seeds, don't expect seeds to be viable. And therefore, lots of companies are a bit sloppy and underperforming, so they sell seeds that are almost dead or completely dead, and there's no control and there's no industry standard. And on the long term, this is going to destroy the industry, because if you plant seed one year, the other year, nothing coming up, you say, natives, it doesn't work, stop using it. So actually, the good players in that market want this quality standard to protect the industry. Another system, very different, is France. They've working very well in the past few years to create like a seed zoning system like in Germany. They've got the problem that the companies that are trying to adhere to this system are variety producers. They produce variety of flowers, like the poppy, the cornflowers. Being variety, they don't care about the origins. What they want is actually have the labels that improve the market value of the products. So it's important to create a strong message to what native means and the importance of origin. But more critically, other countries, like for example, Spain, Italy, but Portugal, Poland, all across the board, there's not such a thing in a native seed market. There's very little legislation in the use of native seeds, and more often than not, is not enforced. So a few months ago, I went in Germany, and we organized this meeting, organized by the German producers, that brought together producers from all over Europe. And the idea is to create this European Native Seed Producers Association, with the goal to support the development of native seed markets across the continent, and provide a common definition of origin and seed quality, define an Europe-wide seed zone, so have the German map actually spread all across Europe, but most importantly is actually after a few years provide the European Parliament in Brussels with the evidences that this is not just a bunch of hippies that want to grow some flowers. This is a serious market with serious implications, not just ecologically, but also economically, especially for rural areas or disadvantaged areas. You can have local production that is wonderful for the local economies. But most importantly, the real question I want to ask is, how are we supposed to restore 15% of degraded landscape if it just 5% of the seeds we're allowed to use is native? Thank you. So, any question? Yes?
In Germany, are the seed zones beginning to cause problems in terms of trading across the zone, maybe with the influence of the climate change or other genetic factors that is stabilising that with the zone system beginning to cause problems?
This seed zoning system started a decade ago. They're going for like 12 years. They started in 2005. Not sure. I know there's lots of study in genetics. I think Anna Bucherova is the main. Anna Bucherova studying, I think, University of Munich. She's doing lots of study with the BWW to see if there's any genetic effect. But so far, seems to be no real evidence at that degree. It's still very up in the air, like it's very debated in academia, this zoning system. And nobody can possibly ever agree with that. Everybody wants the border moved. So. They try to avoid that. More often than not, they try to avoid it, like try to keep within the bigger areas. So what it does, if you see the map, it's got, can you go back to that slide? So if you look at the map of Germany, I'll get faster like this. You've got 8 bigger regions and 22 smaller areas. So the origins are the smaller regions, as you can see now, unfortunately. So you can actually collect from there, produce in the bigger area, and then you sell back then. If there's no material from that area, you can go to the neighboring, but usually the priority gets there. So it's a matter of how many companies there, how big is the market to respond. Yeah. Oh yeah, they're following it. And actually there's even a better system than the German, which has been implemented in Switzerland. They actually take in consideration difference between species. So the map is not the same for all the species. So some species, for example, grasses, they've got like a very little difference genetically and morphologically, they use it across the board. Other species that got more specific differences, like high altitude or some bogs, then they tend to take in consideration like smaller areas. And that's more of a model I think we need to move to in the future when it's practical. The German system? It's complicated. If you watch the video, I'll explain very well, because it's got a system of control, of external control and approval through the government and academia, so it's a bit complex.
So next we have Anna, who's looking at Kunzea robusta and difficulties in direct seeding in New Zealand.
Good afternoon. My name is Ana. I'm a forester from Brazil. I'm currently doing my PhD in New Zealand. And the main focus of my study there is the development of strategies for restore a large area that has burned last year. So this is a picture that shows part of the area. And one of the restoration techniques that I'm testing there is the use of direct seeding of native tree species. So the presentation that I'm bringing here today about the direct seeding of kÄnuka is part of this broader research. First in this presentation is structured in 3 parts. I'll talk firstly about the use of direct seeding in New Zealand. Then I'll talk a little bit about the ecology of kÄnuka, the species focus of this study. And I'll talk about what might constrain the establishment of these species in the field. And finally, I'll bring some of the implications of this research so far. While recent literature mentioned that there is a considerable potential for using direct seeding in New Zealand, the use is still very limited to small areas, more in research scale. And this is probably because there is little research about direct seeding there, and there is still some lack of knowledge in this area. And from a restoration point of view, it's of strategic importance to develop this knowledge in the country, especially because when you compare the direct seeding with the conventional planting of seedlings, there are several advantages for using the direct seeding, especially like usually lower cost than the planting of seedlings. So talking a little bit about the ecology of kÄnuka. KÄnuka is from the Myrtaceae family. It's a tree that reaches 20 to 25 meters, is endemic to New Zealand. The most common species known as kÄnuka there is called Kunzea robusta. It's a pioneer species that is very common after forest clearance or disturbance. And is very common as well after landslides and in rough pasture lands, especially when the soil is exposed, like in this picture in the middle of the slide. And this species is an important species in the forest successional process. So here is just showing how the species is widespread across New Zealand. The black dots shows the areas of occurrence of these species. The yellow square there shows the region where I'm conducting my research. And the satellite image in the right side here is from my study area. So all the black area is the vegetation that has burned, and there is a patch of vegetation that didn't burn. And the red polygon here shows an area dominated by kÄnuka. So all the dark shade green here are crown of kÄnuka trees. So this shows how distinctive is the species in the landscape and how important this species can be for the restoration process of this area. And kÄnuka is a species that presents several advantages from a direct seeding point of view. The trees usually produce huge amounts of seeds that are wind dispersed. The seeds usually present fast germination and high rates of germination in lab conditions and in glass houses as well. So the graphic here is just showing that even after being stored for 14 months, the seeds are still viable and germinate like about 70%, which is quite high germination rate. And so these seeds can be stored for long periods and are unpalatable for mammals. And due to these many advantages, this species has been included in most of the direct seeding trials conducted in New Zealand. And here is a synthesis like from the trials that included these species in the mixture of native species. So the main outcome when trying to direct seed kÄnuka is that the species will fail to establish in the field, even that they present high germination percentage in lab and in glass houses. So this third line here is a research that I have conducted there in that burn area. So I had 100% of failure in all my treatments in the field, even that the seeds germinated 80% in the lab. So from the literature, the first two lines here, they present similar outcomes, except the one in the middle that present one successful treatment that the seedlings established when the topsoil was removed. And failure using direct seeding of kÄnuka is commented between New Zealand researchers, but I guess because they failed, they are not officially reported. So from all these failures emerges the question from this research, which is why this species failed to establish in the field. So aiming to clarify this question, I established the glasshouse trial. So I have collected soil from the same area where I tried to direct seed kÄnuka and that I failed. So I collected the soil, I brought it to the glasshouse, and I sterilized part of the soil, aiming to eliminate any possible soil pathogen. So I have collected soil from under 3 different vegetation types. So I sterilized part of the soil. This resulted in 6 different treatments, 3 vegetation types plus the same vegetation types sterilized. Then I fill up trays with the soil, sown kÄnuka seeds, took it to the glass house where I could control environmental conditions. And 15 days after sowing, the seeds germinated abundantly across all the trays. And 45 days after that, I've counted the number of seedlings. And the results were very clear. Across the sterilized trays, the number of seedlings was greater than across the non-sterilized. The vegetation type didn't influence the results. Here is just a picture exemplifying these trays. And these results suggest that soil pathogen may play an important role in the establishment of the seedlings in the field. And based on literature and on my personal observations, I've established 2 hypotheses that are fungi might be constraining the establishment of the seedlings in the field, or springtails. The population of springtails across the non-sterilized trays was really high. So this result presents a link with the direct seeding of these species in the field and with the natural ecology of these species as well. When the species succeed to establish in topsoil removal or in a condition like this one shown in the picture, when you don't have topsoil, you're supposed to have an area free of soil pathogen. So this matches with these two hypotheses. And to confirm and to test this hypothesis, I'm currently establishing a field trial where I'm testing the effect of insecticide and fungicide in the establishment of these seedlings. I'm testing as well different seedbed treatments, and the aim of this trial is to confirm what might constrain the establishment of kÄnuka seedlings in the field, and in which conditions these seedlings might succeed. So the main implications of this research is that this interaction between soil biota with kÄnuka seedlings establishment is reported here for kÄnuka, but it may be likely to occur with other species and in other countries as well. Establishing a glasshouse trial while doing a direct seeding trial in the field is a good idea, because this trial can help to explain some of the outcomes in the field. And a broader understanding of seed ecology and these first stages of seedlings establishment is an important information to support like the direct seeding approach. And finally, just as a personal observations, failures should be more often reported, as they may be really useful information to improve the direct seeding approach. Thank you.
When you look at kÄnuka stems on South Island in New Zealand, where the seedlings germinate, a very significant part of that ecosystem is a very deep moss layer dominated by hypnum moss. And our observations are that's very critical, not just in the germination of the seedlings in the field, but in their subsequent establishment. And to me, it's rather more likely that it's an environmental, the lack of some sort of environmental protection of the seedlings and their ability to root in the deep layers of moss.
These seedlings that fail to establish in the trays, in the glass house, they fail in very small stage, like microscopic
another question for you later, but I'll find you. Okay. Thank you, Anna. So next we have Alta, who's also staying international. We're looking at seed quality of native seed collected in the zone of Mongolia. Thank you, Alta.
Yeah, thank you. Okay. First of all, thank you so much giving me opportunity to introduce my current research work. So, my name is Alta. I work in an oil mining company in Mongolia, and as a research rehabilitation specialist. So I'm participating in this conference from Mongolia, and most of you maybe know where this country is located. So the Mongolia is located in Asia, neighboring the two countries in China and Russia. And the people have been running nomadic livestock husbandry so many years. But for our last 2 decades, when the democracy was first introduced to Mongolia, development tendencies changed, and now it has transferred to economic form that operates the mining industry. So in this map, we can see lots of the red and blue dots. So those are the lakes. Okay, so unfortunately, those are places the exploration and the mining of the licenses under the mining, under the mineral resources. So at this moment, 3,000 mining licenses have been issued. So the oil company was located in the southern part of Mongolia. It is a copper and gold mining company. So the southern part of Mongolia is with the desert biome of Mongolia. Is a home to wildlife and plant communities with their own unique characteristic, and having established for millennia, depending on specific natural and climatic condition. So you can see here even it is desert, and there are several diversity of habitats. So we are doing the rehabilitation since 2010 for the disturbed lands by the mining activities. So we collaborated with herders to collecting the native seeds. And so we have several challenges. It is in the first time we collected lots of seed but there is no separate condition. So that's why the challenge was is it right to collect a substantial amount of seed when sufficient rain occurs the year, and also under which environment that the seed should be kept, and how to store that seed properly, and how many years or maybe how long we should keep them, and also what kind of policy we should follow further, as we will have no seed bank during the drought season. So that's why So in connection with the rapid development of mining activities in the Gobi region, we used the native plant seeds as good for rehabilitation work. So in order to use rehabilitation and sapling preparation work, we checked the 5 species seed quality, germination rate, and those were Anabasis brevifolia, and Remoria songorica, Salsola passerina, and Eurota ceratoids, and the last one is Zygophyllum xanthoxylon. So we wanted to see the light seed effect of those 5 species seed germination and the temperature affecting the seed germination and also the collecting year affecting the seed germination. So we checked the seed quality, seed germination rate in 5 and 10 gradus, and also checked the daily length is 3 different treatment. So we checked the 10 days and we checked that there are 5 years seed quality, seed germination, and the duration of the lighting had a tendency to influence negatively on seed germination rate, especially the Anabasia brevifolia. And when we checked the temperature, the seed germination rate of Spongia Remoria songorica had a positive correlation with temperature. So in about the seed germination rate of plants and the year of seed collection showed a positive correlation for all species except the Anabasis brevifolia. So finally we could conclude the temperature, lighting, and the year of seed collection had a statistically significant effect on seed germination rates as a result of multi-factor analysis and correlation analysis. So we would like to show our work result to our management level people, and we really need the separate condition of the seed storage. Yep. Thank you so much.
Could you tell me a little about the soils?
The soil is quite a light brown soil in entire the Gobi Desert region.
It is an acid soil, pH?
The pH is more salty, and most of the species growing in the salty soil. Yeah, that's why that we use only the native species for rehabilitation work.
You mentioned that the year of collection impact on germination.
Yes.
Is that, you suggesting there that there might be maternal effects or whatever in a given year that will influence it? Were those predictions from the same populations over time?
Yeah, in the beginning of the collection time, we didn't know much more, and during the collection time, and the seed quality rate was decreasing. And but we collected the seeds just in around the mining site area in the Humboldt-Sum area. Yes.
So can you use that information to predict what might be an appropriate year or an appropriate set of conditions, or do you just sort of know that it's the luck of the draw from year to year?
Yes. Generally in the Gobi Desert areas, it's around 90 millimeter precipitation per year. But some year it's increased in up to 190 millimeter. It happened that just many years. It happened in just 1 or 2 times in the last 30 years. So the seed quality depends on the precipitation. Yeah.
Were your storage conditions very good, or were they just ambient, just shelf storage, or did you have proper storage conditions?
It's just a general condition, and just in ambient. Yes. That's why we are requesting our managers, we need exactly proper the condition for storing the seed.
What length of year, how long of the years collection extended over?
Yes, since 2010 we're collecting the seed. Yes.
I was going to make a comment that we've had the same issues dealing with arid ecosystems where mining practitioners collected seed in different batches, but then only through communication of science and with your managers that eventually you actually can get better storage conditions to help protect the seed that you've collected. It does take quite a while to get people to understand that seed actually
Yes.
needs good conditions in order to increase their longevity. So,
Yeah.
okay, we're slightly ahead of time, which is great. But we move on to Marlien, we're moving into the genetics area, where she's looking at seed, capturing genetic diversity through seed provenancing and collections. I don't know whether you would like to wait, just so that we catch up in time. Yeah. Yes. So it's this one and then up to that.
Okay.
Yeah, twinkling vision until I did a PhD.
Yeah, I think mine was okay until I had kids.
I'll do a public service announcement in the meantime. Just a reminder that the poster sessions are on this afternoon, and to ask really great questions, and then you get a really great drink. Yeah. Perfect sharing rate. So everyone finished on time.
I can make up the time. I've got overtime. Okay, thanks all for coming. I'm from the Royal Botanic Gardens in Sydney, and I'm part of the Restore and Renew team, and I also do some other research of my own, so, which I'll be talking to today. So just as a little starter, I think in terms of successful restoration or successful restored population, if we imagine sort of one species and multiply that, if we start off with some seed, and then they grow, if you're lucky, and then they grow up into individual trees. And if we come back in 100 years, we might find something like this. And that's sort of what we all would kind of aim for, is long-term sustainable populations. So to get this sort of successful restoration, once we've got our mature trees, what we would most like to have is some pollen flow between your individuals, which leads to successful production of fruit and seed, and then subsequently your population grows. Just as a reminder to us all, why do we want to increase our sort of genetic diversity? Well, we've got no pollen flow between our individuals. Well, oops, that wasn't supposed to happen, so go back. I'll not use this. If we've got no pollen flow, but we do have the off chance that we sit with an individual that can self, we might still have some viable offspring that produces flowers, and over time there's more plants, but we're unlucky and a nice pathogen comes in, such as a rust, myrtle rust as an example, and we've got no resistance in our population, so our restored population looks like that. However, if we had a little bit of variation in our population, produce offspring, nasty disease comes along, and we still have some offspring that remains. That's just one of the reasons why we want to try and increase genetic diversity in restored populations. So how do we do this? If we go and look a little bit in the literature, you know, what's good starting material, good restoration, good seed collecting. So seed collecting guidelines, if you go and read the guidelines, suggest, you know, go and collect from healthy adult plants so we can all go into the field and see a plant that looks healthy. So, yeah, that's probably a good thing. We can test for viability and we can look at germination success. But then we come into a little bit of a fuzzy area that's still in the seed collecting guideline. You've got to use seed from local provenance, and you've got to collect seed that represents high levels of genetic diversity. So I don't know about you guys, but I can't go into the field and just collect seed that I know is of high genetic diversity or local provenance, because the data is not available to us. There's simply no empirical data to back up these things. So, lucky for us, got some money to do some work on these things. In terms of local provenance, I think for those of you who went to a previous session, we discussed the project Restore and Renew, which uses genetic, environmental, and evolutionary data to sample across the whole distribution of a species, or kind of currently being New South Wales government. We focus on New South Wales, and then we end up, what we aim for is a free web tool in which you can get an idea of local provenance. With a click of the mouse on the area that you're about to restore, you get a size of your local provenance. But we still sit with this idea that while we're turning local provenance, hopefully over the next few years, into more of a smiley face, we still have this thing, do we actually capture genetic diversity with seed sourcing? So this is one of the questions I'm trying to answer at the moment with the project I'm working on. So it's a sort of a project that adds on to Restore and Renew, which we aim to use Restore and Renew data, then to link it with Restore and Renew data, and then see can we make predictions from species for which we haven't got data for. So just a little bit what we're doing. So the current work on seed versus source genetic diversity. So we're collecting seed from multiple mothers at multiple populations. And we germinate the seed, and unlike most seed projects, we keep track of the mother identity, which adds a lot of work in the lab, but that's okay. So we, as I've just tried to indicate on this little slide, when we germinate the seed, we keep the position of that seed so we know exactly what was that the mother plant that seed came from, so that in the end we can say, if I collected my seed from 10 mother plants, how does my diversity vary from if I collected my seed from one mother plant? And then we can use that information to do more modeling. So we genotype the seedlings and the mothers, and then we estimate some diversity indices. And then hopefully we can repeat this for several species, because I think we've all learned over the past several years that it's unlikely that you can use the one species to say what you should be doing for another species. So in addition to this, we germinate seed at different temperatures, and we keep track of our seed origin or our seed provenance to see if, do seeds from different areas germinate better at different temperatures. This is just something that we have facilities available for, so why not test it? We look at the growth rate versus seed origin, and then we also am lucky enough to be able to do some common garden experiments, growing our seedlings in 2 different climatic regimes to see, does seed origin sort of matter when you actually go and grow something. So this is all in the process of happening, but I do have some exciting results. So just I put this photo up here because it's one of the things that was sort of a byproduct. If we look at this is sort of something that comes out as significant. Germination temperature affects your growth rate. So yes, seed can germinate at quite a high temperature, 35 degrees, which I think most people would agree is tough conditions to be germinating under. But what it looks like is that the actual product of that germination isn't actually very fit. So we've got some coming up data. So that was sort of just something as a surprise that I found while I was doing this. And so just a little bit of output from just looking at the seed germination, seed origin and germination. So we germinated seed. So for this, looking at the germination rates and stuff, in this case, it's a master student's project, and he looked at the mountain seed versus the coastal seed. So this is within the Sydney region. And interesting enough, though, sort of broadly speaking, we find that things do germinate at roughly the same temperatures. We do find differences in the population response to germination temperature. So in the case of both of these species, we've got Acacia suaveolens, a common acacia. I think many of you are familiar with the Australian native species would know it. And then we have Banksy serrata there. Sorry, I should have actually typed out the full names for this. I think I pushed it up a bit. But what we have on there is that at the low temperatures, particularly, we get a difference in response to germination temperatures. What's quite interesting is that, surprising, is that the mountains population from Acacia suaveolens doesn't do the same as the mountain population from Banksia serrata. So there are differences in germination percentage at different temperatures depending on the population or the site that you collected your populations from. Now if we look at growth rate, so just as we were germinating everything and planting them out, we went and measured the root length as an indication of growth rate. And in the case of Acacia suaveolens here, we can see that the coastal populations under all temperatures that we looked at germination grows faster than the mountains population, which again seems to be the opposite for Banksia serrata, that the coastal population grows slower than the mountains one. I don't quite know what to do with those results here. We'll think about it. So now we get to a bit of the genetic data that we've got so far. So you can obviously see that this slide used to be higher up because I've got the names typed out completely here, Acacia suaveolens and Banksia serrata. So what we have on this slide is two principal component analyses of genetic data. And so we make use of DART-seq, which is whole genome scans, and we get SNP data from it. So it's thousands of SNPs across the whole genome of the species. And what we have in the full colors are the mothers, and then in the open circles are the offspring. So contrasting two species, Acacia suaveolens, we find that the space that the whole population, including the seedlings, occupy is fairly narrow. And we've got our 4 populations that we've done so far for Acacia suaveolens, 2 mountains populations, 2 kilometers from each other, and down here we've got the coastal populations. Making a nice smear there. Had a nice macaron for morning tea on the slide, and the coastal populations actually acts as one population, which already gives us some indication of gene flow at different landscapes. So for this species, we get much more gene flow, even though the actual sites that we collected these seeds from were further apart than these 2 sites are from each other. So sort of an interesting, maybe a difference in pollinator, or lack of pollinator possibly. And this is Banksia serrata, which is of course a long-lived species that lives up to about 100 years old, and it also carries the seeds on the plant for a long period until there's a fire. So we still get our individual populations, but the kind of space that the whole population occupies, if you think of a genetic space, is much bigger than what we get in Acacia suaveolens. So if we move on from the principal coordinate analysis in this case, we get to some interesting genetic data just looking at diversity indices. So we've got allelic richness up here, which is a common thing that people often report on, which if we're using SNP data, we've got to start to think a little bit about what does allelic richness mean. So we've got to sort of start to think about these indices that are often used within the conservation and restoration data. Sort of what does it mean? Because if you're working with SNP data, you only have 2 alleles for each one of those markers. So we've got to think about what it means. But what we did pick up here for Acacia suaveolens is that in terms of allelic richness, what we have on here is there we have in dark blue, we've got our parent population. The orange I think we can ignore because it's very biased towards our full sample. And in grey we've got if we collect 1 seedling from 1 mother. And you can see that the allelic richness isn't too much different from our, in general, from our mother population. And then we've got sort of various things that yellow is 2 offsprings, the lighter blue is 3 offsprings per mother. And then in green there we've got 10 offsprings from 1 mother. So surprising is that it's not actually a very big drop in allelic richness. If we look at expected heterozygosity, there's quite a bit of a drop there in what you get if you collect all your seed from 1 parent compared to if you compare it from sort of a 10 or the same as your original source population. But what is very important here is that we can see that expected heterozygosity as well as down here we've got inbreeding coefficient varies quite a lot between populations. And what we found here was that up here in the Blue Mountains, Mount Wilson, we had very low levels of diversity. And if we look at the, so I don't know if this is really related, but when we looked at seed fertility, we had much higher seed infertility up in the mountains than at the coast. And this might be a product of inbreeding. So seed origin could reflect that. So just going into data that we have for Banksy serrata, again we find that in terms of allelic richness, if you collect your same colors here, the green, 10 offspring from 1 mother, grey, 1 offspring from 10 mothers each, that the green gives you lower allelic richness. However, if you looked at, in this case I've put up observed heterozygosity, so I actually sort of feel in many ways it's a better way of looking at the data. But if you look at observed heterozygosity in each one of those populations, so we have got a much bigger data set for Banksy serrata, but it's much, in many populations, it's quite even whether you're collecting from 1 individual or whether you're collecting from 10 individuals, which is a bit of a surprise to us because it's counter. You think, oh, come on, collect from many mothers and you'll be fine. But in terms of your time that you spend in the field and your, so it's sort of your resources available to seed collecting, as well as the amount of seed that you could potentially pick from a site. Just under our permits, we can collect 10% or less of seed available from 1 plant or in a population. So in many ways, maybe you, at some populations, oops, I did it again, you can collect from less individuals and still maintain a high level of diversity. If we look at the inbreeding coefficient, this gives us a little bit of information of what's going on in the species. And Banksy serrata, at this stage, we feel that's highly outcrossing. So you get within 1 mother plant, almost all your fathers come from different places. While in Acacia suaveolens, we think, well, you can often have the same father for multiple seeds. So 2 species doing 2 very different things. Checking time's going okay. So what we've learned so far is not all populations are equal. Allelic richness, mostly higher when collecting from multiple mothers. Heterozygosity, however, doesn't necessarily increase with increase in mothers. Plant mating systems vary across populations, and seed origin may affect fitness under different climatic regimes. So future work with this, so we're hoping to, not hoping, we are doing it at the moment, so we're repeating the same thing across many different species, different mating systems, and different functional groups. And we're starting to look at different genetic diversity indices that might be more appropriate to look at this type of data, such as kinship values, which is this funny little heat map up there. So if you look at your population, what's the sort of probability that you're going to collect seed that are half sibs or full sibs. And then what's your collecting effort versus your gain in diversity? So currently on Restore and Renew we use a model that looks at our genetic data and we predict sort of what's your effort and how much diversity you gain by collecting from more mothers. Well, with the seed genotyping data, we can now use empirical data to look at that. And then hopefully we can, in the end, once we've done a block of species, relate this back to population genetic data of many different species and see if do we need to go and genotype more seedlings to understand this process, or can we use population genetic data from your source population to make predictions about the level of genetic diversity we get from seed populations? And thank you, everybody, for listening.
I do have a question. I'll try and do both at the same time. Just quickly ask, do you, in your model, input the mating system parameters? Because with Banksias, their pollinator is probably quite different to an acacia, which would actually change the level of outcrossing, I guess, that's achieved to that seed. So
yeah, okay.
Inbreeding depression might increase.
So the idea is that we get information about the mating system from the data we get. We all assume, like even with some of the acacias, we assume we know something about the mating system. But currently, that acacia swarmionis, we definitely getting that it can inbreed quite well and it can probably sell. So, but we don't know that, and we make the assumption that something like a Banksia is an outcrosser, but we don't actually have the data to
Yeah, because even if you collect multiple seeds from one tree, because it's outcrossed, you're not going to see that effect.
Yes.
Then if you
cut so hopefully we'll get there. Yes.
Okay. Thank you. So, next we have Melinda, who's looking at choosing appropriate seed sources and the importance of the environment, genetics and demography for actually predicting restoration outcomes.
All right, so we started with European perspective and we'll end the session with that. So at the moment I'm based in Austria, but today I'm going to be talking to you about a collaborative project from a theoretical perspective. So here we're using evolutionary models to ask how do things like genetics, demography, and how do these things affect restoration outcomes. So with being a collaborative project, most of the programming and modelling in this has been done by David Field at the University of Vienna, and we've been doing this in collaboration with a few other theoreticians, including Cezanne Sarakis, Magalhaes Teixeira and Hildegard Ecker, who are either at IST Austria, where I am, or at the Max Planck Institute. So I thought I'd start the session by thinking, when we're thinking about seed sourcing, it's useful to start thinking about populations and how they're distributed in the landscape. So in this illustration here, each of these circles represents a population, where the different colour represents adaptation to a particular environment. And we know that large populations tend to have higher diversity than smaller populations, which lose genetic diversity due to inbreeding and random genetic effects in small populations. And that gene flow among those populations is what? These are all the processes that structure genetic variation. So when we're thinking about restoring, of course, the key question is, well, where do we actually source seeds for restoration? If we're looking at a site that sits here, or do we go for something that's small and local, or do we go for something larger and further away, or particularly from a different environment? And so these are the kind of questions that we wanted to actually put some theory behind and just to look at evolutionary models to see if they could give us any insight into this process. So to think about adaptation, we really need to start thinking about this in a quantitative genetic perspective. So these, because they're basically polygenic traits, or these are traits that are controlled by a really large number of genes. So each gene individually contributes a very, very small effect, but altogether they form the trait value. And so these kind of traits, or polygenic traits, form this normal distribution. So of course the trait that comes to mind immediately when you think about this is just something we're all familiar with, human height. So you've got a normal distribution and you've got a variance around an optimum value. So it's basically stabilising selection that will cause population to be adapted to be around the optimum. But of course you have variation around that. And you have populations that adapt and evolve in response to different environments. So today I'm going to be talking about these kind of the basis of quantitative genetics in terms of seed sourcing in plant systems, but this model is actually really applicable to animal systems as well if you're thinking about restoring a threatened species, because all of these traits of body mass, body size,
Any metabolic processes. For plants, we're talking about flowering time, the amount of flowers, seed size or leaf size, leaf traits. All of these things are polygenic traits. So these are all traits controlled by a really large number of genes. So the thing with seed sourcing when we're thinking about quantitative traits that form the basis of adaptation is we want to make sure that we have a match between the new environment and the source population we're coming from. So this illustration here is from a paper by Carroll. So here we're looking at our trait value, so the distribution that we saw with the optimum around here. And this is the new environment. What we want to try and do with restoration is minimise, so match the genotypes to the new environment, and thinking about all of this in a quantitative genetic perspective. So, but also underlying this too is the problems we know about questions about genetic sources in terms of population size. So, as we were discussing before, we know that large and small populations do different things. So that these small population processes of inbreeding and drift that actually reduce the amount of genetic variation that's in small populations. So when we're thinking about seed sourcing, we need to think about two things here. How do we actually maintain the evolutionary potential of restored populations? And that's all about its genetic base, so how much genetic variation is there for it to be able to adapt and evolve under future environments. And then, of course, is the matching of the environments and patterns of adaptation right now. So they're the two things I want you to take forward when we're continuing through the presentation. So here we're using evolutionary models, and we're going to be asking questions about how does the difference in the trait optimum, which is essentially matching of genetic adaptation, the strength of selection against maladapted individuals, and the number of founders, so that's your demography. How does this all influence our restoration success? Does it actually matter if you're sourcing from a large or a small population? And of course, this question that a lot of people really practically ask on the ground, what is better if you had a small source that was local, but you had a source that was different, slightly different, but it was larger, further away? Where would you actually source? And then to mix or not, what's the outcome when you're actually mixing different seed sources? So these were some of the questions we wanted to explore with our model. So how we actually did this, so we're using evolutionary models. Now, in the absence of a time machine, you can use models to actually look at populations and try and simulate them into the future just to work out what might happen. So to do this, what we actually did within our quantitative genetic model is we had 7 source populations. So you can think about these populations being adapted to different environments. So we could think about a moisture gradient. So this population here might be adapted to really dry conditions, and at the other end of the spectrum, this one might be adapted to really moist conditions. So how we set up the models we took for all of these 7 sources, we took them and then we formed 20 replicate populations. So all of these replicates were basically, we forced in the model that we had the strength of selection for them to be centred on a particular optimum, and we see a variation around that. So the optimum trait value in that environment is given by the blue line. So that's sort of something to think about as we go through here, but we wanted to compare the effect of sourcing from small or large populations. So what we did is we formed, using the model, we had 20 replicates then of very small populations, so 50 individuals, 100 and then 1,000. So the kind of take-home message from this is if you look at them, you can see that for small populations, that distribution wobbles around the optimum. So in our model, we had it set so that they basically should be evolving to the same optimum as the large ones, but small ones don't. And this kind of take-home message here is that small populations aren't necessarily always adapted to their environment because of these chance effects, and because selection tends to be less effective and efficient in small compared to large populations. So just something to keep in mind as we go through the results. So the first question we wanted to ask is this question here, essentially. So you've got the optimum trait value in a new environment. How important is it, and the difference of your sources from that new optimum? How does that affect the actual outcomes that we see? Does that matter about how strong selection is against those maladapted individuals and the number of individuals you put out there in the start? So I'll talk you through these graphs because this is some of the results from our model. So along the x-axis here, we've got the difference in the optimum between the source and the restored population. So essentially, this is the value here. So if it's 0, then essentially your source and the new environment are exactly the same. Then a small value of the difference is where there's a fairly close match, and we're looking up here is where the trait optimums are very different, where you've got a big mismatch between your source and your restored population. And along the y-axis here, we've got the probability or the chance that that population actually establishes. And the different colours here represent the strength of selection. So when selection is 0, it's good that we know our model works because you put individuals out there, there's no selection, so they basically all survive. But as soon as you start putting selection against maladapted individuals, then we see the probability of establishment goes down. But this is actually all related to how the difference between your trait optimums in the source and your restored population. So the larger that difference is, the lower the probability of establishment. And it seems like selection really matters. So when you've got weaker selection, you actually have better restoration success than when you have very strong selection. And this graph here is where we actually started the model out with just 10 individuals. So if we compare different founding numbers here, we find that restoration success improves when you start out with more individuals, as one might expect, so everything is shifted up. But we see the same thing, so you increase the mismatch between the source and the restored population, and that goes down. So your best restoration outcomes are when that difference between the source and your new population is small, and when there's actually weaker selection. So this is illustrated here. So if we take a particular difference from the optimum and we look at founder numbers, so you get a much better success rate with more individuals to start with. And if we now compare two different strengths of selection, 0.5 and 0.1, of course weaker selection, you're seeing greater outcomes. But the bigger the difference you have here, seems that more individuals also tends to buffer you against these genetic effects as well. So this was our general model to start with. But then we wanted to ask the question, well, how does it actually compare if you're sourcing from small or large populations? These are some of the questions we could explore with the model. So with these results here, again, along the x-axis, we've got this difference in optimum between the source, where you're getting from, and the restored population. So remembering down here is where they're fairly tight, and here where you've got a larger mismatch between the source and restored population. And again, we're looking at that in comparison to the probability of establishment. But this time, the three lines represent sourcing from small or large populations. So the red is 50, the green is 100, and the blue is 1,000 individuals. So you can see we definitely see these same trends downwards, so that with the probability of establishment a difference in the optimum. But what you can notice from this is that sourcing from a large population always did better than the small populations. And this is with 20 founders to start with. And it's the same effect we found when you're sourcing with 40 individuals, so more founding individuals, but in this case too, everything is just shifted up with a higher probability of establishment. But the same message is there, that larger populations seem to be doing better than small ones. And this is presumably because you've gotten capturing more genetic variation there. So this leads us on to this third question. We can just explore the data a bit further to ask, okay, well, what happens in this case where you had a small local source, but you had a larger population that might be where the mismatch between the source and your restored population is greater, but it's actually a larger population to start with. Which one of those would you actually choose? Well, we can have a bit of a look at our data to see that. So again, here we're looking at the same figure of the difference in the optimum between your source and your restored population against the probability of establishment. But we're just focusing on this piece here. We're saying, okay, well, these three dots here, we're looking at about a 50% chance of establishing compared to your small population or a large one. But what this actually illustrates is that your large population can be further away from the optimum and still do just as good as a better adapted small population. So this is just giving you a bit of information, a bit of intuition about it, how well, large populations seem to have more variation to be able to cope with a different environment when you compare to when you're a very small population. And the thing to also look at all these graphs is that these small populations seem to have more variation around them too. And I guess that's reflected from those illustrations at the start of the wobble. So sometimes you might get something that's well adapted, others not, because of the chance effects when you're sampling small populations. So the last bit of data is this question about mixing. So what about if you actually mix small sources? Does that actually improve your restoration outcomes? So these graphs are set up in a little bit different way, where the x-axis is the same, so we're looking at this difference in optimum between source and restored population. But this time on the y-axis, we've got the difference in the probability of establishment, because we're comparing two things. We're comparing the performance here of one small versus one large population, and we've got the large minus the small. So that anything that's above this dashed line indicates that that the large source population is performing better. Anything that's below the line means the small source is performing better. So the take-home message from this first comparison is that particularly when you're increasing the distance, so that mismatch between your source and your restored population, the large population is actually performing better. So what happens though if we take some small sources and we actually mix them and we see what the outcome is? So that's what we did here. So we compared mixing two small populations to one large, 4 small to a large, and an 8 small. And we find that when you get to the point of mixing 8 small populations, when you compare them to one large, then they basically perform in a similar way. So it seems like mixing small sources, you're actually getting a greater genetic variation, a greater genetic base for your restoration outcome compared to sampling from, say, one large population. So we've done different combinations of these, of multiple large, multiple small, but this just gives you an idea that mixing can be a good way of capturing that genetic variation. So what's the take-home message from the models? Well, from the results we see that matching populations is important. It's important to consider the environment and the trait optimums, and of course that demography here also can interact with that in terms of more individuals tend to be better. Large populations are often better sources and are likely to be better sources, but in the absence of large populations, mixing lots of small ones can actually give you a similar benefit. So this is some really kind of the new results that we've got from this model. There's actually more we'd really, really like to do with this. The first of these is that at the moment small populations are inbreeding, so they're more likely to be related individuals in there. But those inbred individuals are not penalised by inbreeding depression, so this is something we have to put into our model. And probably what we expect the outcome to be is that'll just penalise small populations more. So it might expect that difference between small and large to become greater, but I don't know. We'll have to see when those models come out. We want to test some scenarios around composite provenancing. So what happens if we actually combine from different environments? What's the outcome? And of course, one of the biggest questions we want to know, and something that's really nice for models to look at because we can't go backwards and forwards in time, is like, well, what happened with these restored populations? What if we actually change the environment on them? And then we expect them to evolve. Do they have the capacity to evolve in changing environments, as you might expect under climate change or any other scenarios of rapid environmental change? So that's something we would like to continue and do in the future. And then, of course, this leads us, well, this is a model, and of course models play a really important role in being able to test and try and understand the underlying processes that are going on, but they can also provide some predictions that at least we can test in the short term by some kind of adaptive management model if we put some restoration plantings out there with different mixes, and then at least we can monitor them over the short and hopefully the long term to see if they actually adhere with any of the predictions we might see. And of course, when we're talking about quantitative genetic traits, it means going out there and measuring trait values, but there's a lot of innovation now in terms of a really efficient means of actually collecting trait values that might mean that that's actually possible, and that we can use models as well of the environment to link trait values to environment and use that as a prediction. So thanks for your attention, and I'd be happy to take any questions.